Equipment: brine shrimp egg, 6 beaker, marker pen, water bath, and refrigerator at following temperature 15, 20, 25, 30, and 35 degrees centigrade, thermometer, sea salt, weighing scale, measuring cylinder, spatula, dechlorinated stringing rod, graph paper, scissors, magnifying glass, forceps, pipette, light sources, equipment list, seed, 5 petri dishes, marker pen, water bath, thermometer, cotton, 50 centimeter square measuring cylinder, distilled water. Ethics: when handling living organism for scientific research and learning, it's important. Treat them with the care, minimize the potential harm. When using the organism experimentally as a student, you have a responsibility to gain as much advancement in the knowledge and learning as possible from them. The availability of organism you could use the experiment should be considered, since those easier to obtain will make increase the practicality of the research. Method 1: brine shrimp. Use the marker and pen to mark 5 beaker with following temperature: 15 degrees centigrade, 20 degrees centigrade, 25 degrees centigrade, 30, 35 degrees centigrade. Use the weighing scale and spatula to measure the weight, weigh out 2 gram of sea salt into each beaker, followed by the 100 centimeter cubic dechlorinated water, means chlorine should not be there. Stir the mixture with the stirring rod in order to dissolve the salt, means we have to create the marine environment because shrimps are found in the marine. Dampen some paper with a salt solution and place a pinch of brine shrimp egg on the paper. Use the magnifying glass to separate and count the egg until they have roughly 40 on the paper. Cut the paper around the egg so that the size of the paper is smaller than the area of 5 beaker prepared. Then place the piece of the paper egg side down into the beakers, means all different beaker where the temperature is different. Allow the 2 to 3 minutes for the egg to fall off the paper and into the beaker, and finally removing the paper from the water with tweezers or forceps. Repeat the step 2, 3, 4 for remaining 4 beaker. Place the marked beaker
Sir,
into, yeah.
are we going to use the same paper and dip
No,
it in different beakers or different paper?
no. Just what this paper is just saying that you have taken a piece of the paper, right? Suppose that you have these 4 beaker, right? So what you have to do after spreading the egg, you have to cut down this paper
Okay.
in the pieces. Every piece should be placed on a beaker. Got it?
Okay.
And you have to cut down the paper like this way so that the size of the paper should be equal to the surface area top the bottom of the beaker. Got it?
Yes.
Okay, and it should be upside down.
Okay.
So place the marked beaker into water bath. Place the marked beaker into water bath and refrigerators that correspond the temperature written on them level of 24 hours. Prepare the 6th and last beaker as described in step 2. Remove the first beaker and use the bench lamp or light source to shine light into the beaker, causing the brine shrimp that has to swim in the surface. Remove the first beaker and use the bench lamp. Okay, fine. Light source, bench lamp, that study lamp, and source of the light so that the beaker, causing the brine shrimp to have has and swim into the surface. Use the pipette to remove the shrimp one at a time and placing them into empty 6 beaker as you count them. Record the number of the brine shrimp that have hatched at that temperature as a result of table as a result table and place the hatch shrimp a location instructed by your teacher. Repeat the step 8 and 9, the whole thing, in reviewing 4 baker, right? So in the nutshell, we will try to know at what is the optimum temperature. So the optimum temperature should be that at which the maximum shrimp eggs will be hatched. Got it?
Yes.
So that was the first experiment. So here we
Sir,
have
can you scroll up? I need to ask a question. Yeah. Point number 6. It says place the marked beakers in water baths and
first
refrigerators
place,
that.
yes, first place the marked beaker into water bath and then refrigerator.
Yeah, refrigerator of the same temperature of the test tube of
No,
the
no, no,
beaker.
no. The refrigerator, different refrigerator will have different temperatures.
Yeah, so like the beaker has 15 degrees temperature.
Exactly. That
So
should be
if I
separate
put,
separately.
yeah, but should I put it in the refrigerator that has the same temperature?
No, 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 no. You cannot put that. First, you have to repeat, do this experiment at 15 degrees centigrade. Then you have to repeat this experiment with the 20 degree, then 30 degree, like this way.
Okay,
Or
but
if
what
or if
what
you
I'm
want to do the experiment simultaneously, you require 6 fridges.
okay.
You cannot keep all these beaker on the same fridge at the same time.
No. I'm not saying we have to keep all of them in the same fridge.
Yeah.
I'm saying yes, it's gonna be placed in different refrigerator, but for example, the beaker's temperature is 25. I
Okay.
should put it in a refrigerator of 25 temperature, right? Or
Right.
should the refrigerator be of another temperature?
No, no, no. It should be at the 25 only,
Okay.
because the fridge will decide the temperature of the that beaker.
Okay.
So that's how we have to control. We have to control the temperature with the help of fridge. Then we will
Oh,
come to know that which is the optimum temperature.
okay.
Got it?
Yes.
Yes. Any more question?
No.
Method 2, seedlings. Use the marker pen to label 5 petri dishes with the following temperature: 15 degree, 20 degree, 25 degree, 30 degree, 35 degrees centigrade. Mold 1 centimeter layer of the cotton wool into the bottom of each petri dishes and place 20 seedlings, right, on the petri dish. Using the tweezers and forceps, spread the seedling out so that they evenly distributed across the dishes, right. Use the measuring cylinder to slowly and add 30 centimeter cubic of the water, right. So mold 1 centimeter layer of the cotton and wool to the bottom of petri dishes, okay, and place the 20 seedling on the petri dishes using the tweezer or forceps, right. Spread the seedling out so that they evenly distributed across the dishes, right. Use the measuring cylinder slowly and add 30 centimeter cubic of water. 30 centimeter cubic of water means 30 uh, ml of the water, right, to each dishes. Then place each of the marked petri dishes in their corresponding temperature and warmth. Every 24 hours, add more distilled water to each of dishes so that the seedling and the cotton wool does not dry out. Be sure to add the same volume of the water each dish, right? If you are adding the extra water to petri dish, then the amount of the water should be same for the all petri. After 5 days, remove the petri dishes from the water bath and refrigerator and count the number of the seeds that have germinated for each temperature according to the result of the suitable environment. From this experiment, we are trying to prove that what is the impact of the temperature on the germination of the seed, right? And above experiment, we were trying to prove that what is the optimum temperature for shrimp, what should be the optimum temperature for shrimp to get hatched, right? So here, in whatever the temperature, in which temperature, whichever the temperature, the seedlings are more seedling will germinate, that will be the optimum temperature. A graph can be plotted to the temperature against the number of the shrimp hatch and seedling. Can you plot this graph?
Sir, temperature will be on the x-axis?
Temperature should be, no, you can you can take it the temperature at the y-axis, not x-axis.
Why?
Adjustment. Hold on. Let me try it. Because the axis is defined in the respect of the time. Time always should be in the x-axis. But for the rest, there is no access hard and fast rule. So yes, temperature should be in this. Like
15, 20.
you can eat 10, 20, 10, 20, 30, 40, 45, right? Like this way. And here the temperature should be again 15, 20, 25, 30. Number of the seedlings, suppose that their germination is 30 only. Then the germination at this temperature is 40. Then here again 21, right? Here again 10, right? So the graph will be like this way. It's simple.
So why would the graph look like this?
Because if suppose that the germination of seed optimum temperature is 25 degrees centigrade. If the temperature will be less than this one, then the rate of the germination will be low. If the temperature is more than this one, then the temperature will be, then the germination rate will be low, right? So germination rate will be maximum at this temperature, right? So definitely
germination
the
is
graph
maximum at 25?
Right? Sorry?
The germination the germination optimum temperature is 25?
No, that's just I'm giving you example. It's not like that. It depends on the seed to seed actually, right? Because in North Indian plain the germination should be taken at the 20 degrees centigrade, 25 degrees centigrade, right? And it also depends on the what type of the seed we are growing. But for the European country, those are adapted for the European environment. That should be the different. So that depend on the seed to seed, right?
Okay.
We cannot say that what is the optimum temperature, but as per my view, it should be around 20 to 25 degrees centigrade for most of the plant. Okay.
Okay.
Liquid. Look. The spilling could cause the surface of slippery and leading to the precaution, wipe any liquid and all that. Wipe soon as they occur if they spill out, right? Seedling, brine shrimp, potential allergic reaction, wear the gloves to handle them. Glassware, cut sharp object, take care of while handling the glass object. Keep away from the edges of the desk. Lamp, it could get hot if the lift off for the prolonged period of the time. Turn off after the use. Take care of when using the avoid the skin contact. So go through with it. Is it okay? Or if you have any doubt, you can ask me, right?
No, I don't have any doubt.
Okay. 13. Investigate the rate of growth of microorganisms in a liquid culture, taking into account the safe and ethical use of organs. A calorimeter or light sensor with a data logger can use the measure the rate of the growth of the microorganism like yeast. As a yeast population grow, the broth they live and they become more cloudy due to the presence of more microorganisms. This increases in the turbidiness means less light passes through the culture. So by monitoring the light passing through the culture, across 24-hour period, their growth can be estimated, right? A calorimeter or light sensor with a data logger can be used to measure the rate of the growth of the microorganism. Okay, like this. The yeast population they live, they become more cloudy due to presence of more microbes. The increase in the turbidity means the less light passes through the culture. So monitoring the light passing through the culture across the 24-hour period, growth can be. Is that clear?
Yes.
Right. Equipment, bench disinfectant, Bunsen burner, calorimeter, 0.5 percentage of glucose solution, cuvettes, measuring cylinder, conical flask, pipette, yeast culture, magnetic stirrer, weighing scale. Before starting the experiment, disinfectant the workbench. This helps to prevent the contamination when preparing the yeast culture. That is true. Fill the 500 centimeter cubic conical flask with 250 centimeter cubic of the glucose solution, and then weight out the 1.25 gram of the yeast and add the flask along the magnetic string plea that enable you constantly and eventually stirring the culture. Right. So a conical flask with glucose solution. Right. Uh, the conical flask should be 5. This is the volume of conical flask. This is the volume of glucose solution. Right. Why the glucose solution? Because this is the best medium for the growth of the yeast. Right. Weight out the 1.25 gram of the yeast. So that is the dry yeast which we have to weight out, and then we have to add it to flask and then stir. Place the flask on magnetic stirrer base stopper flask with some cotton wool. Secure the covering or with the aluminum foil. Why? So that the external organism should not enter through that. Right? Flask can be incubated at room depth so that the infection could not go.
So what is magnetic stirring, please?
Magnetic stirring, that is a like rod like thing which with the help of that we mix something. Like whenever you mix sugar syrup into the water and you stir it, so that kind of the stirring, nothing else.
Okay.
Measuring growth matter method 1, calorimetry, with the help of calorimetry. Calorimetry, how does it work? We know. Use a pipette to fill cuvette with a 0.5% glucose solution and take calorimetry reading. So that the reference calorimetry is of the equipment. Have you seen that?
No.
That is like microscope, nothing else. The only difference is that calorimetry will throw some light, right? It have some inbuilt bulb-like structure thing which project the light, and on the basis of opacity of the substance, it tells the population growth. Reading the reference of absorbance of the calorimetry to 0. Once the yeast sample has been prepared, use this sterile pipette to transfer 4 centimeter cubic of the culture into a cuvette and take a absorbance reading, recording the age of the culture in the minutes and absorbance in the table. Right. We have to measure how many minutes before you have started the making culture, right? And then you have to record the population. Repeat the step 1 and 2 for the ages 30 minutes. So first we will measure the culture in 30 minutes, then 1 hour, then 90 minutes, then 2 hours, then 5 hours, then 8 hours, then 11 hours, right? So we have to take these different readings, right? And we know that after every reading, the population will increase because the yeast will multiply by the process of budding, right? Measuring the growth method 1, light sensor and data logger. Right? Set up the apparatus shown, right? This is the culture of yeast. This is magnetic stirrer, right? that steer this one, and cool light source, then data logger that will tell with the On the basis of the opacity, it will tell the what is the density. Leave the data logger continuously record absorbance of light for 12 hours, right? So
Sir, why
in the
12 hours?
sorry,
Why 12 hours?
12 hours, because here what we see that here 30 minutes, 1 hour, 10 hours, 2 minutes. So after at different hours, we'll take the reading. Got it?
Okay.
The same thing. We are doing the same thing, but with the different method. Here we are using calorimetry, and here we are using data logger, right?
Okay.
We are doing the same experiment with two different methods. Risk liquid spillage that cause the surface slippery and leading to accident. Wipe up liquid spillage as soon as they occur. Put lids, bottles, put them away from the use. Glassware means they can cut us, like yeast may cause the allergic reaction. Contamination of the yeast with other microorganisms, right? Wear the gloves, avoid the skin contact and all that. Use the aseptic technique. Okay. Disinfectant that may cause the allergic reaction. Avoid the skin contact. Allow to take them some workbench. The table absorbance, right? We are reading like at the 0.5, 1, 1.5, 2, 2.5. So we'll take the reading. The shape of the graph would show the experiment. Why? Because first the yeast will grow rapidly, and after that, when resources will become what? Resource will become optimum. Remember,
Yes.
the graph can never be like logistic graph. It will be S-shaped, sigmoid growth. After when the maximum limit will be reached, the number of the population will become constant. When the resource will become optimum, when the glucose will become optimum, water will become optimum.
So after like how many hours pass by when the optimum resource will, optimum?
It depends on the culture, right?
Okay.
Like it is given on the 2 hours. So
Okay.
that depend on the experiment. That depend on the yeast. That depend on the temperature. Number of the things, right? That may be optimum. May reach at the 5, reach at the 2, reach at the 1.5. It depends. That is not fixed. Okay.
Okay.
So this was the experiment. Go through with it. If you have asked me, if you have any doubt.
Okay. So what is magnetic stir base?
Look, they have shown. Give me just. This plate, it will keep on shaking the water so that the water get mixed evenly. Glucose remains evenly so that the yeast do not settle at the bottom. Got it?
Okay.
It will keep on moving this way. Magnetic steering.
Okay. So this setup is for the same experiment.
Exactly this setup. So here we are using the two different method to observe the rate of reproduction or rate of the growth of the yeast.
Okay, so this is another method, and the cuvette and calorimeter is a different method.
Another method, right? But almost the average reading should be both for the same.
Okay.
The
So in
graph
this
should be
method
same
we
for the
use
both.
so in this method we use calorimetry to see the absorbance reading,
Right.
and in the other one we use the cardboard box to prevent outside light. The other apparatus.
This one.
The one
Sensor
Okay.
data logger.
Okay.
But almost the graph should be same if the time
Okay.
difference is same.
Okay. No, sir, I don't have any doubts.
Okay. 14. Investigate the effect of different antibiotic on bacteria. The effectiveness of different antibiotic on the specific bacteria can be investigated into following practical antibiotic kill the bacteria or slow the growth and their effectiveness, and this will be shown by the clear area produced around the sample of each antibiotic. What are the independent variables? Type of antibiotic use, dependent variable, area of inhibition zone, right? Equipment list, 5 different type of the antibiotics, agar plate, filter paper cut, 6 equal size disc, sterile forceps, disinfectant, known bacterial culture, sterile pipette, sterile water, sterile plastic, and spreader, Bunsen burner, ruler, and tape. Before starting the experiment, disinfectant workbench. This helped to prevent the contamination when preparing the bacterial culture. Mark on the agar plate. Uh, Mark on the agar plate the date, your initials, and the bacteria being grown. Also mark evenly across the plate 6 letters A, F, and show where the plate have the different antibiotics. So mark the other plate, the date. Okay, so here we have to mark the date and your initial of the bacteria being grown. E. coli bacteria, like which bacteria we are growing, E. coli bacteria we are growing. Also mark evenly across the plate 6 letters A, B, C, D here, and so where the place different antibiotic list, right? Okay. Use the Bunsen burner and flame bottle of the neck of bacterial flask, causing the air rise and carry away the undesired airborne microorganism. And then use the sterile pipette to transfer the 2 centimeter cubic of the liquid broth on the other place. Use the sterile plastic spreader to evenly distribute the bacteria across agar jelly. Fine. Take the first filter paper disc and use the forceps completely submerge into first antibiotic for the 10 seconds. Slightly lift the lid of the other plate and gentle allow you to quickly transfer the disc in the other jelly. Did you got this point or not?
Yes.
Repeat the step 5 and 4. This one, 5 and 4. As well as a control disc, soap, sterile water, sterilizing forceps between each antibiotic by holding them and Bunsen burner flame. Right. Tape the lid of the 4 point, making secure enough, but also allowing the oxygen to enter the bacteria. They respire aerobically. Right. So we have to make some holes so that the air should go inside for aerobic bacteria. Then incubate the disc at 25 centigrade for 48 hours. We have to keep the disc in the incubator. Incubator is a box-like structure, microwave oven-like structure, where we maintain the temperature. Why 25? Because this is the best temperature for the growth of the bacteria. Disinfectant the work surface again and wash your hand. After 48 hours have passed, use the ruler to measure the diameter of inhibition zone, clear zone, to create around the disc. To minimize contamination, have a lid removed from the agar plate. Work near the Bunsen burner so that air from the heating air should be sterile. Bacteria, if not handled safely, appropriately, users could become infected. Right. So store them bacteria safely. Minimize the time spent. Disinfectant agar jelly could cause the skin irritation. Right. Antibiotic A, B, C, D, E. Diameter of the clear zone. Area in the clear zone. Means where the clear zone will be more, means that antibiotic will be more effective.
Okay, so if there's more clear zone, then the antibiotic is more effective?
Obviously, because the clear zone will be more, means the more bacteria are not there. I mean, the less bacteria are there, right? Because what is the function of antibiotic? Antibiotic function is to kill the bacteria. So
Yeah.
those antibiotics that will be more effective, that will kill more bacteria, means there should be more clear zone. Got it?
Yes. So if I had to make a graph of this, how would I make it?
Simple. Here you will get the area, right? You can do one thing. Uh, you can take antibiotic. A, B, C, D. Right. Here you can take clear zone in millimeter cubic or centimeter cubic, cubic, right? 10 cubic, 20 cubic, 30 cubic, 40 cubic. And here you can make bar or whatever you want to make. You can make graph.
Sir, when should I make a bar graph and when should I make a line graph?
It depends whatever the question is asking. If question is not asking you, um, then you are free to make any of the graph, whatever you want.
No, but like, well, how would I understand which one is more suitable? Bar or
Normally, we draw the bar graph when it comes about like volume library. When we have to represent the volume, weight, then we use this. Otherwise, generally we use the line graph.
okay. So what about the question that I asked, the graph question?
Sorry, what about the?
The graph question that I asked in the last class.
That I have forwarded to physics teacher, but actually he's busy with the classes, so I haven't got any response from his end. Right, but I forward to him.
Well, my exam is on 1st June.
Exam is on 12th June.
No, 1st.
Okay, 1st June. Okay,
Yeah.
okay. So only 6 days left.
Yes. So I think I prepared for my unit 5 much better than my unit 4.
Sorry, you.
I think I prepared for my unit 5 exam much better than my unit 4 exam, which is on the 1st June.
I didn't got you. What exactly you want to convey me?
No, I'm saying
Yeah.
on 1st June, I have my exam, unit 4, topic 5 and 6.
Okay, topic 5 and 6. Fine.
Yeah. And on 5th June, I have unit 5 exam, which is coordination and
Fine.
topic
Control.
sound.
Okay.
Okay. So I paid more attention to unit 5, so I'm more prepared for that, the 5th June exam. And I'm not that ready for my first exam.
Why?
Because I paid more attention to my Unit 5 exam.
But in the both of the exam, they are the equally mark fetching.
Yeah, equal marks.
So focus on the both. You have ample of time.
Yeah, I am focusing.
Okay. So then just first prepare for this one, first June exam, right?
Okay.
Ah, and if you have any doubt, you can ask me, right?
Okay.
Yeah. So try to get maximum marks.